This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 57 for November 4th, 2009. Hi, everybody. I'm your host, Vincent Racaniello, and today we have a special and slightly different TWiV for you. Several weeks ago, I visited my local high school, Scotch Plains Fanwood High School, and taught virology to the biology classes there. My motivation to do this was twofold. First, my older son Aiden, who you might recall as a guest on TWiV number 7, had asked me to visit his class and talk about viruses. In addition, on an earlier TWiV, a listener had suggested that I try to engage high school students on the podcast by including some of them. So I took that advice and scheduled a day of teaching high school students about viruses. I contacted my son's biology teacher, Miss Dooley, who asked if I would also teach other classes besides hers. In the end, I spoke with 7 different classes of AP and accelerated biology students. I gave them a general overview about viruses, what they are, how they propagate, and how we study them. Then I spoke briefly about the 2009 H5n1 influenza virus strain and the vaccine against it. A PDF of my PowerPoint presentation is available for download at twiv.tv. At the end of each period, I had the students ask questions about viruses, which I then recorded along with my answers. Those sessions form the main part of this podcast. I think you'll be very interested to hear what great questions the students asked. Their curiosity comes through clearly, and their concerns about the H5n1 vaccine mirror those that I've seen in the general population. I also spent a few moments speaking with the biology teacher who helped arrange the day, Miss Dooley. At the end of the day, I was exhausted and had newfound respect for high school teachers. I also felt that I probably should have spoken less and engaged the students more. I'll keep those lessons in mind in a few years when my other children are in high school and I return to teach them about viruses. Please stay tuned after the question and answer session for a few brief announcements and some reader email. First name?
Scott.
What's your question, Scott?
I was wondering if you could speak to us. There's a lot of people in the news, famous people, that are rallying against vaccination, in particular MMR, and getting children vaccinated. And the latest data is that the data for that was not so great. So I was wondering if you could just kind of clear that up for anybody that might have a misconception about the MMR vaccination in kids.
It's a tough question. So Scott is asking me why famous people are saying we shouldn't receive vaccines. What this originates from is a report in the late 90s by a British physician. His name was Andrew Wakefield, who studied about 10 children with autism, which is a developmental disorder of children. It's manifested very early in life. And he claimed that shortly after these children received the MMR vaccine, they developed autism. MMR is measles, mumps, rubella. It's combined, 3 viruses combined into one vaccine. So he said what happened in these kids, they got injected with the vaccine virus went to their intestines, it grew in their intestines, and made their intestines permeable so that the children were taking up toxins from the environment, and that's what caused the autism. So to make a long story short, his study was flawed, and subsequently 10 different epidemiological studies were done to see if there was any correlation between immunization with MMR and the onset of autism, and there was none. So in an epidemiological study, what you do is you study many, many kids. You have half of them get the vaccine, half of them don't, and you compare the rate of autism in both populations. And if the vaccine causes 1 in 15,000 cases of autism, you will see it. And of course, the proponents of not vaccinating say it causes autism in all kids, and it's never detected. So this was taken up by famous people in the press. And I mean, of course, the problem is parents whose children are autistic want to know why they became that way. We don't understand autism at the moment, so it was very convenient to blame a vaccine. But I can tell you that there is zero evidence that these vaccines or any other vaccines were responsible for autism. And unfortunately, the result is that, especially in the UK, immunization rates with MMR went way down. There were outbreaks of measles, and measles can kill you. There were a couple of deaths in England. Totally preventable. But because there was a false idea that these vaccines were dangerous, this happened. Now, to this day, immunization rates have never recovered in the UK. No vaccine causes autism. There's zero evidence. They're all pretty safe. No vaccine is totally safe. There are always side effects. If you get the flu vaccine, you get swelling, you may get a little fever, but they don't cause autism, for sure. That's the short answer. Hey, what's your name?
Ryan?
Hey, Ryan, question.
You said how the HIV virus can never be eradicated. So how did they eradicate the smallpox virus and polio in the beginning of the 1900s if we can't do it with HIV?
It's a great question. So the problem with HIV is that the viral RNA, when the virus infects cells, it gets converted to DNA, and that DNA gets integrated into your cells. So you can get rid of all the virus in a person by treating them with antiviral drugs. And that's why many people live for 10 years or more with AIDS now, because the antivirals make you disease-free. But the DNA is still in your genome, so you can never get rid of that. And from time to time, it can produce new virus particles. So that kind of lifestyle isn't present for smallpox or polio. Once you get rid of those viruses from a host, they're gone. There's no DNA left behind in the host. And that's the real problem with HIV, that we don't know how to get rid of that DNA from our cells. People are working on it, but until we figure it out, can't eradicate it. All right, I have a question for you. You have a question? Okay. Hey, what's your name?
Hi there, Lynn Canfield. Swine flu, is it as scary as the media is making it to be?
What do you think? Is it scary? No? Who's going to get immunized against swine flu? Nobody? One person, the teacher. You know, I taught a class in emerging infections last week. It's graduate students, and they had the same response. Nobody was going to get the vaccine. Why don't you want to get it? Just curious. You don't have the time? Your parents don't want you to get it? No reason.
Unsure about the results.
Unsure?
Don't a lot more people die from normal flu than from swine flu anyway, statistically?
It's about the same. The fatality rate between swine flu and seasonal flu is about the same.
I don't feel like I need to get vaccinated for normal flu, so why would I get vaccinated for swine flu?
So the problem is that in the U.S., between 25 and 50,000 people die a year from influenza. And if everyone were immunized, we wouldn't have that. The problem is when not everyone is immunized, the virus can still spread, and so it hits people that are not able to fight off the disease. But anyway, your question, is it as scary as the press makes it out to be? Absolutely not. As you said, it's no more serious than seasonal flu. What was the scariest flu ever? Does anyone know? 1918. All right, this killed almost 100 million people, like 20% of the world's population. It was a particularly virulent virus, but there's no evidence that this is anywhere near that. So this is more like seasonal flu. The press has hyped it, and I think the result is everyone doesn't believe it, and no one's going to get immunized. So we have all kinds of problems with the press, and to top it off, the press doesn't understand viruses at all, and they get it wrong. And believe me, I talk to them as much as I can to straighten it out, but I can't talk to everyone. So this isn't terribly scary. It's no more serious than seasonal flu. But this year, nobody is immune to this virus, and that's the problem. In seasonal influenza, a lot of people have immunity from the previous year. But this year, there is no immunity, so many, many more people are going to be infected. And if the fatality rate is 1 in 25,000, which is what it seems to be, a lot of people will die. Mostly older people, very young kids, and pregnant women seem to be the most susceptible. So my view is if everyone got immunized, if everyone were immunized, there would be no spread of the virus. You could save all those lives. So here's the thing. Vaccination is sort of an altruistic thing. You have to do it for other people, not just for yourself. And I'm not sure that that is something that we're all comfortable with. But anyway, I got my shot last week, and I think you need to do it because it's really good for everyone. Any other questions? Anybody want to be a virologist? No? Maybe? Okay, my email is up on the first slide. I can give you clues about how to do it.
There are different viruses that are more susceptible to medications than others. So for like HIV, there are some strains that we have better treatment for than we do with others. I had also read a story a few years ago about kind of applying natural selection to this process where we can treat for some so that the ones that were more susceptible will come back in greater numbers, and then we could really hammer those with the drugs that we have. So it's kind of like giving people a treatment for a period of time and then taking them off the treatment for a period of time to increase the counts of the virus that's more susceptible to the drugs, and then whacking them really hard with the drugs and kind of working off and on with a cycle like that. I was wondering if you could speak to us about that.
All right, so what he's referring to is a treatment of a number of years ago. So we have a lot of antivirals against HIV. These are drugs that you can take which will block replication. And a number of years ago, the idea, so one of the problems is that you get resistance to these drugs very quickly when you take them. And about 10 years ago, we were treating with one drug at a time, and within a couple of weeks, you get resistance. So the idea was to treat with drug and then stop, let the viruses grow back, and then treat again. And the idea would be that the susceptible viruses would grow out, and then you could knock them off. Unfortunately, this is a great way to get resistance, so we don't do that anymore. It was a bad idea, and it's not done. What we do now is we treat people with 3 drugs at a time. It's called triple therapy, and it takes a couple of years to get resistance to all 3 drugs. And when you do get resistance, you just change the combination. So, Magic Johnson has been living now over 10 years with AIDS because every 2 years they switch his combination of drugs. Now we have about 20 or so drugs, right? So you got 7 different combinations you can use theoretically, and then you're out of luck. But hopefully in the meantime, new drugs are developed, and there are always new ones. So with disease the extent of HIV, there's always new drugs being developed. So we identify new targets in the virus, new enzymes that we can inhibit, and pharmaceutical companies are always making new ones. So now the regimen is to use combinations and not do it's called interrupted treatment. Yeah. Anything else? Yep.
Oh, hi, my name is Patty Davis. My question is, how would you compare viruses to prions?
Viruses and prions. You guys know what a prion is? A prion is a protein that seems to be infectious. So there are prion diseases like mad cow disease, where you could eat contaminated beef. It has a protein in it that will make its way into your brain and cause neurological diseases. So when prions were first discovered, the only people who paid attention to the prion scientists were virologists, because we were the only ones that appreciated that this could be. So a prion has no nucleic acid. It's just a protein, and it's infectious. So it's different from a virus, because all of our viruses that I've told you about have nucleic acid genomes. Prion is just a protein, and what happens is this protein goes into your cell, and it makes other proteins like it misfold, and they become disease-causing. So if you ingest a prion, it will induce a disease in you by making your proteins misfold. So it just goes to show that you don't need nucleic acid to be infectious. A protein by itself can do it. So if you have heard, sometimes beef gets contaminated with prions and it can't be sold, this is what the cause is, these infectious proteins. But one more student question. It'd be really great for the podcast, because I'm trying to do nationwide involvement of students in virology, and if they hear you guys, then they're going to do it more. Yes, what's your name?
I'm Bill.
Okay.
And you said viruses can be spread through contact. So like how long, like let's say I sneezed on a doorknob, how long would it take until like a guy couldn't get it?
Okay. So how long? So influenza virus, for example, about a couple of days it will last on a doorknob. So they've done studies in daycare centers where you have all these hard plastic toys, and the virus can last a few days on the toys. Of course, it varies from virus to virus, but flu is the most important, most likely. So you have to be very careful. You should wash your hands a lot. When you're in a school like this is a great place to spread flu. As soon as school resumed this fall, flu rates shot up, right? Because in the summer everybody's scattered, there's not much interaction. But this is the great setting because you're in one room, and you don't have to sneeze like I showed you to make great aerosols. I am making an aerosol right now talking. You're all making little aerosols breathing, and thus viruses are coming out. So if one of you has flu in a room like this, you can easily infect everyone else. So it's not just the touching, but you have to remember that if you're ill, you should stay home. But I'm sure that's not a problem with you guys. And if you're touching in a school, lots of things, you should wash your hands before you eat lunch or touch your face. It's really important. All right, thanks a lot. This episode of This Week in Virology is brought to you by Citrix, the makers of GoToMyPC. Here's the problem. A typical work week used to be Monday through Friday, 9 to 5. But boy, have times changed. Most of us are spending more and more hours stuck at the office. You're missing out on important family events. You're putting off your personal life. What's the solution? Well, I recommend GoToMyPC to access your office computer from home. You don't have to be at the office to get your work done. You can work from home early in the morning or late at night or on the weekend, after you've done the things you want or need to do. Our listeners can try GoToMyPC free for 30 days. That's a month of unlimited remote access free. For this special offer, you must visit www.gotomypc.com/podcast. That's gotomypc.com/podcast for a free trial. And we thank Citrix for their support of This Week in Virology. Okay, what's your first name?
Emma.
Emma, what's your question?
Can viruses only grow in white blood cells, or does it?
Okay. Can viruses only grow in white blood cells? So HIV can. There are a few other viruses that can. But other viruses can grow in virtually every kind of cell. So influenza virus, for example, doesn't grow in white blood cells, but it grows in the cells of your respiratory tract. So every virus has sort of a different cell that it likes to infect. And sometimes, so that's one of the things we try and study in virology, what controls that. Another question.
Lynn Canfield, a follow-up to that question is there any cells that viruses don't like?
For certain viruses, there are some cells that they don't like. For example, poliovirus loves your nerve cells and it infects them, and that's why you get paralyzed, but it doesn't replicate in your respiratory tract. For every given virus, there's a very specific pattern of cells it likes and it doesn't like. And we try and understand why that's so, because it may give us ways to block infection. So if we can figure out why a virus doesn't like a particular cell, we could then design a way to inhibit infection. Another question. We ask you, are you guys going to get influenza vaccine this year? If you are, raise your hand. Nobody's, one hand, okay. Yes, what's your name?
Alyssa. If you already got tested for the influenza, if you're already sick, can you get it again?
So if you've already had influenza, can you get it again?
Yeah.
Yes, and not in 1 year though. If you had influenza in the past few months, you won't get it for another year. But influenza is a little bit different from most viruses because it changes every year. That's why you can get infected every year. So from year to year, the strain changes enough to infect you again. Okay, so if you had flu over the summer, you're probably okay for this year. A lot of people ask me, if I've had flu this year, should I get the vaccine? And I tell them, well, you don't know, there are several strains of flu circulating, and one strain doesn't protect you against the other. So I tell them, you might as well get the vaccine. There's no downside to it. Any other questions? So we had just one person getting the vaccine in this class? Two? You've already had it? And that was the seasonal vaccine, right? Are you going to go back and get the swine flu? You don't know. Okay. It's very interesting because in all the classes I've asked, the S rate is very low. I don't know. Are you guys going to get immunized? Teachers? You are? You? Okay. You know, you should, because it protects everyone. Even though you may think flu is not a big deal, if everyone in the world got immunized, which is impossible, we don't have enough vaccine, but if everyone theoretically did, you would stop the spread of the virus. But we can't eliminate it because it infects all these other animals which we don't immunize. So, one more question. I really need it for my podcast. I want to hear high school kids asking questions. People ask me all the time on my podcast, why don't you get high school kids involved? And I say, all right, I'm going to do it. So one more. There's got to be one question you like. Just one. You have a question? No? You don't want to know anything about viruses? I can tell you anything. I have answers to everything. Well, right. What's your name?
Haley. Are there viruses all over us right now, or?
What a great question. You know, there are bacteria all over you. You have more bacterial cells on and in you than actual human cells. Someone asked me that in my podcast recently, and nobody's looked on your skin anyway. In the gut, you have lots of viruses. They're mostly viruses of bacteria, and there are plenty of those. There are probably other kinds of viruses in you, but on your skin, I'll bet there's a whole family of viruses right on your skin, and probably that's being done right now. This is a good question. Okay. All right. Thanks very much, guys.
Yeah, thank you.
Okay. What's your name?
Ben.
Okay. What's your question?
You said that I believe 5% of our DNA is made up of virus DNA. Right. Could you explain the significance of that and what it really means for us?
Okay, so this is an interesting question. Some years ago, the sequence of the human genome was determined. So all the millions of bases. And then shortly after that, the sequence of the chimpanzee genome was also sequenced. And we're 95% identical. And the 5% difference between humans and chimps is all virus DNA integrated in our genomes. And I saw a student in my department once with a T-shirt that said 95% chimp. And that's true, because 95% of our DNA is chimp DNA. So what is the 5% DNA doing there? These are all what we call ancient viruses. They apparently have been with us for years. All of them are defective. They don't make infectious virus. They're just pieces of DNA in our genomes. So we don't know what the significance of them is, but the fact that they're the main difference between us and chimps suggests that at one time in our evolution, maybe many, many years ago, they played some role in our development. Maybe they made us able to stand upright or some way distinguished us from chimps. So there are a number of species besides us that have DNAs integrated in our genomes. We don't know what they do. Very interesting problem. Any other questions? A couple more. I need material for my podcast. Yes, good. What's your name?
Alexis.
Okay, what's your question?
And I want to know when you're getting vaccinated for the flu or any type of flu, what's the difference between the nasal spray and the shot?
Good question. Nasal spray is a infectious virus. It's produced actually in eggs. It's grown in eggs as opposed to grown in cells because it's very easy to grow a lot of virus in eggs. And that virus, actually, you inhale it and it grows in the cells of your respiratory tract. It mimics a real influenza infection. And many people feel that a vaccine that mimics a real infection is the best kind of vaccine. The other vaccine, which is injected, is also grown in eggs, but it is inactivated with chemicals. So it is not infectious. It cannot reproduce in you. And it also works pretty well, but it's not as effective as the live vaccine. I think the infectious vaccine that you inhale is the vaccine of the future, and eventually will phase out the injected vaccine. But right now, 90% of all the vaccine against flu that's produced is this egg-grown, non-infectious production. They both protect you pretty well, but the live one is a little better. Okay. Viruses. You must have a question about. Oh, let me ask you a question. So raise your hand if you're going to get the swine flu vaccine. This is the best response so far. Previous classes, 1 or 2 people, and I had a class 2 weeks ago. It's called, this class I teach to graduate students is called Emerging Infections, because new viruses are always emerging, and nobody in that class was going to get the vaccine. Okay, one more. Yes, you have a question? What's your name?
Hershita. Do you know how, in I don't remember which year it was, SARS, the virus, what was exactly about that? Because I always had, I have wrong information for that.
Okay.
How were you guys able to, find out the vaccine for it, besides, breaking the code?
Okay, SARS emerged in China a number of years ago, and what happened was there's a live meat market in Hong Kong, or in the provinces, where animals are brought in, they're butchered right in the open market in stalls, and people buy them. And these animals are contaminated with SARS coronavirus. And some of the handlers got infected, and then it spread globally, essentially. There are thousands of infections and a number of deaths. Within a year, the spread was stopped because all this live animal butchering was stopped in China. So they stopped the infusion of virus, and then through containment, the infection spread was stopped. So there was no vaccine ever developed, but it was just through containment that we stopped the spread. But this, sorry, say it.
Because someone told me there was a vaccine for it, and I was like, I don't.
No, there's no vaccine for SARS. In fact, it's gone now, and we don't really understand why it's gone, but it's no longer around, so there's no reason to make a vaccine. It's still in the animals in China. We think the actual source is bats. And in the field, the bats infect these other animals that are then brought to the live food market. And we think we interrupted the chain, and that's why it's no longer in people. But it may come back again. We're not sure. Now, the interesting thing about SARS is that within a few weeks of this discovery of this new respiratory disease, we got samples from people who are infected. And there's a lab at Columbia where they can determine the identity of the virus within a day. And there's a whole new field of virology now. It's called virus discovery. Wherever there's a new outbreak of a virus disease, you go, you get samples, you bring them to your lab, and you can immediately sequence them and discover what they are. And what we're learning is that all of the new infections that we get, for example, SARS, all the new influenza infections, Ebola, Marburg, they all come from animals. And as our population grows and impinges on animals, so we make big farms, we destroy the forests, we have more and more contact with wild animals and people, we're getting all their viruses. So they're not really new viruses. They've been there forever. It's just that we're contacting the animals more. We have global transportation and we're picking them up. Another question? One more? One more. It has to be one virus question. I want to make you famous. Come on, you'll be on iTunes. Your sweet voices. Has to be one. You have a question?
No, I'm just thinking. Why is it that, you have you say that bats transfer SARS or something like that? Why is it that they don't all die from it? Or something like that.
Great question. Why don't bats or any animals that have these viruses, why aren't they killed? Exactly. No answer. Somehow these viruses evolve with the bats or whatever the natural animal reservoir is, and they're fine. They get along just fine. And we don't study this enough, because if we understand why viruses wouldn't hurt their hosts, maybe we could, intervene in human infections and do the same thing. So there are many animals, even mosquitoes, that have virus infections. They don't get sick. We don't understand it. And it's a growing area of research to try and understand why these animals don't get infected. It's a good question. Okay, that's great. Thanks very much. We're not done yet. You want to answer?
Lynn Canfield again. Safety in your lab. How do you prevent, once you bring those viruses into your lab, how do you prevent them from leaving? And how do you keep yourself safe?
Okay, so the viruses we work with either are not very serious or we've been immunized. So we work on poliovirus. We've all been immunized against polio. We also work on viruses that cause the common cold. There's no vaccine, but they're relatively mild infections. Nevertheless, we have to be careful. We work in these laminar flow hoods. We wear gloves. We wear lab coats. We wear protective glasses. We are very careful. about our waste. We don't dispose of infectious waste except in the receptacles that are designated for it. And in those laminar flow hoods, we have containers of bleach. 2% bleach is actually the best way to inactivate most viruses. So when we pipette a virus, we put the pipette in the bleach to inactivate the virus. So you actually have to be trained when you come to the lab to work in these ways. It's very important because you don't want to infect yourself or anybody else. And we make new viruses in the lab often, and we don't want to release those into the environment, of course, so we have to contain everything. Anything else? Okay, what's your name?
I'm Jamie. Why is it that, like, the swine flu is different than the regular flu?
Okay. Why is swine flu different from regular?
Why is it so much worse?
So the new swine flu strain came from pigs, and it's a combination virus of viruses in pigs and humans and birds. And none of us have immunity to this virus, so we've never been infected with anything like it, and that's why so many people are going to be infected globally. It's actually, the disease is not much more serious than regular flu. It's the same kind of disease, the same respiratory disease, same transmission. Mortality rate is about 1 in 25,000. So of every 25,000 people that get infected, 1 will die. But the simple fact that none of us can fight this virus, none of us have immunity, means that more people are going to get very sick. Otherwise, it's very similar. Now, there are some strains of flu that are very different. For example, you may have heard of Avian Flu H5n1, which is extremely lethal, but it only spreads from birds to people. It doesn't spread from person to person, and it hasn't acquired global spreading capabilities. And the flu of 1918 was also quite lethal, and that's very different. So this new H1N1, the only bad part is that nobody's immune, so there will just be more infections. Okay, and that's why we should all be immunized to prevent spread. Now, I've found that many people think they don't want to be immunized because it's not a serious disease, but if you're immunized, you protect other people. You cut down the ability of the virus to spread. So if we had, say, 80 or 90% of the world immunized, the virus would not be able to spread. There would be no issue. But we never can achieve that rate of immunization because people don't think it's so important. All right? All right, thank you. Okay, go ahead.
You know how, well, everyone, in order to be in schools, they have to get vaccines, and for certain viruses. What do you think of people who are who believe that there should be vaccine choice, that not everyone who goes into public schools should have these vaccines?
Well, in order for vaccines to work, everybody has to take them, or the large majority of the population has to take them. And if they're safe, which they are, then I don't see why you shouldn't have to take them. So, you need a core of vaccinations in order to go to school. And I realize there's a lot of opposition that people want a choice, but I think they want that because they don't understand how they work or the fact that they're safe. It's a really important question because it's a lot of debate about it. What do you think?
I'm not sure. I think everyone should have it, but I see everyone, even on TV, saying how the benefits don't outweigh the problems and the risks. So
no, I think TV is wrong. They need to talk to virologists and epidemiologists, and if they did, they would understand that the benefit far outweighs the risk. Here's an example. In England, people stopped getting the measles vaccine a number of years ago because they thought it caused autism, which turned out to be absolutely untrue. But the consequence was a lot of kids got measles, and 4 of them died. So you can protect against that kind of lethality with these vaccines, and they're totally safe. So I think mandatory immunization is the only way to ensure that over 90% of the population gets the vaccine. So while, it may not be the ideal situation, I think we have to do it.
Okay, thank you.
All right, that's a good question. Your name?
I'm Cindy, and I'm wondering, I've got a daughter who's over in the Peace Corps in West Africa right now. I'm wondering if there are things I could be telling her to do. I mean, she's in a very remote, very primitive environment. Is there anything she can be doing just to boil and filter her water? So,
well, there you have risk for these hemorrhagic fevers,
I know.
Ebola, Marburg, and so forth. And so she should stay away from caves because that's the primary risk, but she's probably not going into caves. The problem is if there's someone who is ill That's often the way they're transmitted. Early on in the illness, it isn't recognized that it's a hemorrhagic fever, a viral hemorrhagic fever, and often the people who treat the sick individuals die. So she has to be very careful with anyone with some exotic illness to stay away from them. Unfortunately, there are no vaccines yet, but eventually for people who do work in these at-risk areas, there will have to be some developed. That's the best thing she could do, stay away from caves and be careful about sick individuals. Right? And if she goes out into the country, be very careful about what kind of wildlife she encounters.
Okay,
Yes.
that sounds all good to me.
That's interesting. There are a lot of people that are working there, obviously, and they should be aware of the risks. They have to know that these are serious, but rare, but serious infections. But there are ways that you can reduce the risk.
Okay.
Yeah.
Well, thank you.
Okay. What's your name?
Michael.
Okay, what's your question?
When the virus goes into the cell, how does the virus know to like attach to it?
Good question. So viruses don't actually know anything, right? They're just chemicals. Everything is sort of accidental. It's evolved over years to work. It's a chemical reaction. So, sometimes you can mix two chemicals together and they react, and other times you mix them and nothing happens. It's sort of the same thing with a virus. The most important thing is that on the surface of the cell, there has to be a dock for the virus to attach to. If there's no dock, if there's no protein, the virus will not infect. So HIV only will infect cells with a particular receptor, and that receptor is only on white blood cells. And other viruses have similar requirements. So the main thing is to have that entry port on the surface. Okay? Good question. Okay, next. Yes, your name?
Christian.
Okay.
What came first, the virus or a living cell?
Fabulous. That's great. I have no idea. There are two schools of thought. It's a good question. No, they're all good. I mean, if a virus needs a cell, right, then the cell had to come first. But I can imagine, many, many years ago, we think there was this soup on Earth of organic matter. And that's where we think life emerged. I can imagine just a viral nucleic acid being able to reproduce in that kind of soup. So I think that the viruses could have come first, and they may be reproduced not in a cell, but in some kind of organic broth, if you will. Unfortunately, we'll never know, because there's no fossils of viruses. A lot of other animals, we discovered dinosaurs from fossils, but we don't have a fossil record for viruses, unfortunately. But so we can't answer your very good question, but either one, in my opinion. It's a really good question. What's your name?
Alex. Once the virus attaches to the cell, is there anything the cell can do to try and fight it off?
Absolutely. Fabulous question. Okay. There are many things, and in fact, as we research viruses, we learn about these natural things that cells can do to fight off infections. And the reason we want to know is so that we could use them ourselves. So cells have defenses inside them, many different kinds of defenses that can fight off viruses. They have enzymes that can chop up the nucleic acid of the virus. They have proteins that can bind to the viruses and neutralize them. In fact, that's why we don't get sick most of the time. So there are viruses here that are infecting us, but they're stopped at these early stages. So we call these innate defenses because they're always there, and they're very efficient. Now, if those fail, then you have another kind of defense which takes about 10 days to develop, which is antibodies. And those kick in, and they prevent the infection as well. So yes, there are very early stages of defense, many different kinds. We're just learning about them. And then if they fail, you have others as well. You guys have the best questions so far today. Yeah, your name?
Scott. What makes some viruses stronger than others? Why is the HIV virus so much more harmful to the body than a common cold?
Why are some viruses more pathogenic than others? I think it's a complicated question. Part of it has to do with what kind of cells the virus infects. So HIV is unique because it infects actually the cells that are involved in your defense. So the question we just talked about, your body's defense mechanisms. Those lymphocytes are part of that. So HIV infects those cells, and it basically destroys your defense system. So that's why it's very serious. You actually don't die of HIV infection. You die of other microbes that infect you, common microbes that we have no problem clearing. But because HIV is destroying the defense cells, you die from them. Whereas a common cold infects your lung cells. They're not involved in defense, so it's much milder. Okay. Yeah, name?
Andrew. So we know that viruses are good at replicating, but can they go extinct?
Can viruses be eradicated by us or just naturally?
Just naturally, like die out like animals.
Yes. So if a virus only grows in a certain kind of animal and that animal becomes extinct, then yes, the virus is gone, unless it could adapt to grow. And there's some examples of animals that have gone extinct over the years, and the viruses that infect them are gone as well. So that's one way of getting rid of human viruses. If we go extinct, there will be no more viruses, but then, of course, we won't be around either. So there was another question in here, right? Did you have a question?
Paige, how do you get, once a virus is inside of you, how do you get rid of it? Because if it's alive, then how do you, kill it if it's not alive?
It's good. Okay. If it's alive, if it's not alive, you can't kill it. Well, it's a chemical, so you have to get rid of the chemical if you look at it that way. So your body's defenses have many ways of getting rid of it. So you could, so these early defenses that we talked about before, they can chop up the nucleic acid of the virus before it gets going. That happens within the cell. Let's say those defenses fail and the cell makes virus, and now you have viruses floating around in your body. Your body will make proteins that bind the virus and destroy them. So there are many, many different mechanisms that can get rid of them. So the idea of killing, this is problematic, because if they're not alive, how can you kill them? Exactly. So I call it infectious and non-infectious to get around that. But I always make a mistake, and I'll say, for example, a live virus vaccine. So some vaccines are non-infectious and some are infectious. And we used to call them live vaccines or killed vaccines. And people always say, well, how can you kill them if they're not living? So it's a semantic issue, but that's a good question. Okay, anything else? Yeah.
Do I have to say my name again?
No, I know who you are. Alex.
Okay. How does medicine kill the disease or get rid of the disease?
So how do drugs get rid of virus infections? Okay, so if you remember that movie I showed, there are very distinct steps in the way a virus grows. It attaches to a cell. We have drugs that block that attachment. So we have drugs that will bind the proteins on the cell, and they prevent the virus from attaching. If the virus gets in, remember those enzymes that copy the nucleic acid, we have drugs that block those. So we have drugs that will block almost every stage of viral replication. And there are many different stages in that life cycle that you can block. So that's how they work.
They just put a wall in front of it, or?
That's one way of looking at it. So the receptor, you could imagine putting a wall in front so the virus can't get to it, right? And the same with the enzymes. The enzymes have a little concavity on the surface that the virus has to fit in, and this drug will fill it up and block it, basically. Yeah. So for HIV, as I said, we have 20 drugs that are used in people to prevent infection. And they all have different, we call them targets. Some of them hit the receptor, some of them hit the enzyme that makes nucleic acid. And I don't know if you remember, when the protein was being made, there was an enzyme that was cutting it up. There are also drugs that inhibit that enzyme. And you could look at them as putting up a wall. That's a good way of describing it. The virus can no longer do its thing.
Josh, how do viruses become immune to medication and stuff?
Good. How do viruses become immune, or we call it drug resistance? So let's say you design a drug that makes a wall on the receptor protein on the surface. It turns out that wall can be blocked by just one small change in the receptor on the cell. So very few changes in the protein are needed to prevent the drugs from binding to their target. So in fact, that's very common in antivirals. If you just use one antiviral, you get very rapidly viruses which have slight changes and they're resistant to it. So now for influenza, you may know TWiV is a very commonly used antiviral. but you very quickly get resistance, and by next year all the viruses will be resistant to that. But there are very small changes in the targets is all you need. Yep, name?
Chandler, what's the purpose of the dock on the cell? Because if it's the only, like, if it's an easy access for the virus to get in, then what's the purpose of it?
Good question. So the dock has its own function in the cell's life cycle. So they have to be there in order for the cell to live. And the virus happens to have co-opted it. But exactly, yes. Thanks, everybody. What's your name?
Aiden.
What's your question?
How often does a virus mutate?
How often does a virus mutate? You see, you all always have good questions. You shouldn't be scared. Every question I've had, every time a virus copies its nucleic acid, it mutates. Every time. So when you take an RNA and make another RNA from it, there are a couple of mutations already. And so in the course of a replication cycle where there are many, many steps of copying, you have many, many mutations. And that's, of course, why you get resistance to antiviral drugs because of this. Okay, next. What's your first name?
Taylor.
Okay.
How long can a virus be dormant, like without a host?
So how long would it survive on a table? Depends on the virus. So the ones with membranes around them are less stable. So influenza virus would probably last no more than a couple of days. Of course, if it's very hot, it won't last even that long. But there are other viruses that are very stable. For example, poliovirus is a virus that you ingest. It goes through your your intestinal tract. It's very stable. It will last in the environment for up to a year. So there's a big range depending on the virus. Next question. I need like 3 more. Okay, what's your first name?
Amanda. Do certain viruses die at certain temperatures?
Yes. Some, like influenza, are inactivated at maybe 75, 85 degrees Fahrenheit. And the poliovirus that I just talked about, 80 degrees or higher. And that's, again, because of the structure. So viruses that look like icosahedra have a very stable structure, and you can heat them quite high. Whereas the ones with the membrane tends to break when you heat it. That's a good question. Okay, what's your first name?
Ross, you said that your opinion was that viruses are not alive, but if they're not alive, then how can they die?
Good question. Well, so I don't think they can even die. They can be inactivated. So you can look at a virus as a chemical that can do certain things, and then you can prevent that activity by heating it or treating it with another chemical. I've had many people say that to me. How can you say viruses are not living if you can kill them? So it's sort of a semantic thing, right? You don't really kill them. You inactivate their infectivity. But I should tell you, my opinion about viruses not being living is not shared by everybody. So let me just take a poll. Who thinks viruses are living? And you probably have good arguments for it. And even my most learned colleagues, very smart people, some of them think that they're living and not. You can argue both ways. So it's certainly not, if I made a list of facts about viruses, probably their not being alive wouldn't really be a fact because it's more of an opinion. Did you have a question? You? I'm going to go through every one of you. You already did. Any questions? It's easy to ask a question. And you're going to be on iTunes forever. It's more embarrassing than cool, right? Do you have a question? No? You ever have a virus infection? Have you ever had one? Sure. Good answer. How many of you are going to get the swine flu vaccine? Just raise your hands if you're going to get it. You already got it? You're not going to get it. Not yet. You will. Nobody. It's very low uptake of this vaccine. Okay, we need 2 more questions. You got 6 minutes. Okay, first name.
Colleen, how do you think vaccinations, do you agree with how they relate to autism and other diseases?
Okay, do I think that vaccines cause autism or any other problems? And no, they don't. There have been a lot of very careful and extensive studies. There have been at least 12 different studies done. So here's how you figure out if a vaccine causes autism or anything else. you take a very large population of children, tens of thousands of children if you can find them, and you look at those that get a vaccine and those that don't, and you compare the rates of autism in both populations. And this is a field called epidemiology, and there are people trained to do that who are really good at math, and they can figure out if by comparing two populations there's an effect. So this has been done in 10 or 12 different studies with millions of kids, and there's no difference in the rate of autism in those that receive the vaccine or don't. And that's the best way to answer that question, and there's just no association. At one point some people felt that there was, but they were based on faulty science and bad studies, and they've all been disproven. Unfortunately, once the public hears this, they don't believe the opposite answer. So I think there is no evidence for involvement of vaccines in autism or in any negative aspect. There are always side effects of vaccines, but the benefits of preventing these infectious diseases far outweigh those side effects. Okay. Did you have a question too? Okay. First name?
Mary, can you get a vaccine, a virus from a plant to an animal or from an animal to a plant?
It's interesting. Can a virus of an animal infect a plant or vice versa? There isn't any evidence so far, but in theory, I don't see why it couldn't happen. There's some evidence that viruses have gone from plants to honeybees, for example, which makes sense because bees hang around plants. There's nothing documented for, say, animals. But this is a good question. People have asked me that on the podcast before. The way viruses get into plants is very different from the way they get into our cells. So remember, in our cells, they have to bind to a receptor and get taken in. But plants have this wall of very tough material before their plasma membranes. And so the only way viruses can get through that is if an insect pokes a hole in the plant, or, say, a piece of farm equipment breaks the crop, and then the virus can get in. So they tend to be very different viruses from human viruses. But so I would say there's no evidence, but why not? Any other questions? You must have always wanted to know something about viruses. Did you give him a question to ask? Good. Okay. What's your first name?
Jordan. Can viruses destroy non-living material?
What is that, from a video game or something? Can viruses destroy non-living material? Well, a virus cannot replicate in anything that's non-living, so the answer is no. So if you have a corpse, a dead animal, a virus will not grow in it. The cells have to be metabolically active. Yeah. Anything else? Did you enjoy this? Was it useful? Did you learn something? All right. Thank you then. That's the end of my part. You have another one? You're Aiden, right?
If you have a lot of viruses in one spot, does it affect do they have like a pH or something like that?
If you have a lot of viruses, does it change the pH?
Yeah.
So if you put viruses in a solution, no, actually, they won't affect the pH of solution, but pH can affect viruses. So viruses with membranes, like influenza virus, if you have them in a buffer and then you lower the pH, the viruses will actually fuse, because that's what happens when they get into a cell. There's a whole pathway of things getting into cells, which I'm sure you haven't learned about yet, but which involves membrane fusion, and that involves lowering of pH. You have another one? Okay.
Does radiation kill viruses?
Oh, absolutely. Radiation will kill viruses, will inactivate the nucleic acid. It's a common way of inactivating viruses. More questions? Good. That's great. You have the most so far. You guys. First name?
Ryan. How many viruses come out of a sneeze when you sneeze?
So each droplet, can have up to 20,000 viruses in it. So next, when you sneeze, if you have a cold, you try and contain the sneeze. Did you have a question? Yes.
What's your name? First name? Emily. Could two completely unrelated viruses infect a cell at the same time?
Very good question. Yes, probably, and it's been shown in the lab many times. Some viruses will interfere with each other, but others can coexist. So, can you make a new virus that way? By combining bits of the other two. We've never seen that, but I'll bet it can happen. Yeah. Anything else? All right. Thank you. All right, what's your first name?
Billy.
Okay, good.
How do you get viruses in the container so people don't get sick, like in your offices and everything?
So how do we prevent ourselves from getting infected? So we work in those hoods. So the virus is always being worked with behind the glass, and the airflow keeps the virus from coming out. And then if we have it in a tube, we make sure the tube is sealed before we take it out. But let's say we use a piece of a pipette or something and it's contaminated. Before it leaves the hood, it gets soaked in bleach. Bleach is a great way to inactivate viruses, and then we put it in a red bag, we call it, where it gets burned and autoclaved. All right, who's next? Do you have a question? No? You? All right. Excuse me. What's your first name?
Kim. In the video before, when you were saying how viruses like spread, how long does that actually take?
So spreading within a person or from person to person, say?
Like spreading within a person. How long until?
Well, it depends on the virus. So let's say HIV within a week or so can establish a significant infection in you. Influenza in 1 or 2 days. So the minute you have a virus entering your respiratory tract, 1 or 2 days later, you start to feel ill. So every virus has a different incubation period, and they all have different lengths of infection. HIV can go for 10, 20 years, and influenza is over in a week. Good question. Yeah. Yep. Okay. First name?
Jesse. Why are colds so common?
Why are colds so common? Okay. Good question. First of all, great. There are probably 500 different known viruses that cause the common cold. So you could get infected by one, and then you're immune to that one, but there are 499 others that would infect you. And they're basically, all these viruses are spread all around the world, so wherever you go, you're going to encounter most of them. So even if you got 1 or 2 a year in your lifetime, you'd never go through all 500, and that's why they keep you. Why do they clog up your nose? Okay, so these viruses go into the respiratory tract, as I showed you here. That's a very good question. Well, they're all good. Let's show you the respiratory tract. So they replicate, the viruses grow in these ciliated cells, and your body wants to get rid of the viruses, so they start to mount a defense. And part of the defense is to bring a lot of cells into this area, and part of it is to make a lot of mucus and fluids. And it's basically your immune response that's clogging your nose up. So the virus isn't doing that, but your body's response to the infection is doing that. Excellent. What's your name?
I'm Trisha. How come, like, when you get, like, the flu or something, or you're sick, how come you can't give it to, like, your dog?
Okay. So there are strains of influenza that infect dogs, but they're different from the human strains. So dogs get flu from other dogs, not from people. It probably has to do with a number of things. Probably the receptor on the cell isn't right on the dog versus the human. And probably some other things as well. But you can give flu to birds and pigs, but not dogs, right? Yep.
Alex, there are different kinds of flus, like the stomach flu, the normal flu, swine flu. What do they all have in common that characterize them as flus?
Okay, so influenza or flu is a respiratory tract disease. When you have stomach flu, as people call it, it's a different virus. It's usually a gastroenteritis virus. So let's go back to flu. Swine flu, seasonal flu, it's all the same disease. It's just a different virus. And the severity is about the same. First name?
Andrew. How do you get viruses into a contained area so that you can examine them?
Very good. Sort of, that's a good question. Here's what we do. We have a lab at Columbia where they are called the Virus Discovery Lab. And they get samples from all over the world from people who are really sick. So last year there was an outbreak of a disease in Africa. Nobody could figure out what it was. So they got saliva and sputum specimens. They put them in a container. They put it in many, many layers of containers. It's put on a special airplane where everyone knows there's infectious material in it. It's flown to the airport. The lab people go out, pick it up, and they drive it right to the laboratory. There's a special elevator for just specimens that goes right up to a lab where it has the highest containment. And then they can study it in there. And what they do when they get these very infectious specimens, they immediately destroy any infectivity with a chemical, and then they just look at whatever nucleic acids are left. So this is increasingly important to do because now we're getting more and more examples of exotic infections where we have to study them in labs halfway around the world. So it's a very interesting process. Others, yes.
Deirdre, since flu vaccinations are made like a year in advance, can they really be trusted to treat particular strains of the flu because there's a different one every year?
Right. So they're pretty good about the timing. So the vaccine that we're using now for the pandemic swine flu, the strain was picked in April, and that's just about enough time to make a vaccine, and it hasn't changed sufficiently. Now, in a typical flu year where we have seasonal flu, not a pandemic strain, influenza is seasonal in the northern hemisphere. So in this country, it peaks. It starts in November, peaks in January, February, and then by May is over. We then pick the strain that circulates down south, and we use that the next year. Most of the time, it's pretty good, but it's not always a perfect match, and we lose that. But as we make better ways to speed up that process, it will improve. Thank you very much. Your first name, please.
I'm Kristen.
Okay.
How do you feel about the swine flu vaccine?
I think the swine flu vaccine is great, and everybody should get it because it will prevent the 1 in 25,000 people from dying. So I don't mind about getting flu myself, but I would like to stop anyone else from getting it. If everyone took the vaccine, there would be no spread of influenza. So I think everyone should get it, but unfortunately not everyone will. As you can see from the response in this class, very few people will. There was a question in the back. Syracuse. You have a question?
Yeah,
Hang on, let me get to you. What's your first name?
Jordan.
Okay, Jordan.
Is the what's with the AIDS vaccine? Is that real?
No, there's no AIDS vaccine, not yet. There are lots being tested, and they've all failed. There are antiviral drugs, so if you have AIDS, you can live a complete life forever, or as long as humans live, but there's no vaccine yet. Okay? Next. Who's next? You have 30 years of virology experience. You can tap. You can ask any question. You'll probably get a good answer. What have you always wanted to know about viruses? Yes.
Meredith, how do we get better when we get a virus?
Okay. So most people do recover from viral infections. Not everybody, but most. How do you get better? So we have this great defense system. It has 3 stages. And the first stage blocks most infections before the virus even gets into cells. The second acts once the virus gets into the cells and eliminates most of those. And then the third stage is when you make antibodies against a virus when it goes too far. And those antibodies are just big proteins from the cell that can attach to the virus and prevent them from infecting. And eventually those are removed from you. So in most infections, let's take flu. Flu is a typical what we call acute infection. It's over very quickly. You get infected, you get symptoms, but during that time when you have symptoms, your body is actually mounting your defenses. And eventually they get rid of all the virus with these antibodies binding to the virus particles, and that's when you get better. So your defense systems are essential in clearing a virus infection. And that's why if you have AIDS or an organ transplant, you can't get rid of virus infections. And people with AIDS don't die of AIDS, they die of other microbes infecting them. Next. Just need a couple more. Hey, your day is almost over, and then you're done. So a couple more is all I need.
Why do some people get sick more than others? Is it genetic?
Yes, it is. It is absolutely well. So the largest component is genetic, and we don't understand what parts of our genes are responsible for that. But there's certainly people who are more susceptible to infections, and those that are not. And many labs are trying to understand what's going on. But there's another component, and that is how you take care of yourself. If you are well fed and rested and do all these good things, you have a higher resistance to viral infection. If you're undernourished, if you don't get enough sleep, if you have a bad lifestyle, you're more likely to get. And one of the biggest risk factors for respiratory infections is smoking. You increase your risk enormously because when you smoke, you destroy these nice ciliated cells lining your respiratory tract, and that gives you predisposition. So genetics and lifestyle are the two main components. Syracuse, do you have another question?
Yes.
All right, I'll be there. Sorry, what was your real name? Jordan.
How far does a sneeze actually travel?
All right, so those pictures, it's just like a foot or two, but the smallest drops can go the length of this room. So the big drops fall right down here, and they don't do much good. But when you sneeze, you make a whole range of drop sizes. And believe it or not, people actually study this in the lab. They have people sneeze and they measure the drops. And the smallest ones, which you really can't see, can travel across the room. So if you sneeze, you can infect someone over there. Okay, first name.
Brian. You said you studied viruses in your lab. Where did you get the viruses from?
This is a question I hoped someone would ask. Where do they come from? So there are two sources of viruses. So I worked in someone else's lab, and I got them from that person. I took them to my own lab. So that's one source. You get it from lab to lab. Or you can get them from people who are infected. So now people get flu. I had a cold a few weeks ago. I took a little respiratory sample and I put it in cells, and I'm growing my virus out in it. So you can get it either from a freezer, someone's freezer, or from a fresh specimen. Good question. Yes. First name?
Zoe. How come, since like our skin is made of like a lot of cells, how come like viruses don't affect our skin as much as it like affects our inside?
Good question. Our skin is actually covered with a dead layer of cells. The outer layer is dead, so no virus can infect them. It's a great barrier. The only way viruses can get through is by an insect bite, a scratch, a dog bite. So, the skin is our biggest organ, and you would think it's the most susceptible, but because the outer layer is dead, it's great protection. But anything that's exposed, mucosal surfaces, our eyes, for example, respiratory and alimentary tract, places where gases and chemicals have to diffuse, that's where the viruses typically go. So they're all great questions. Okay, first name.
Kristen, and how are different strains of viruses caused?
Why do they exist, for example? Okay, so the one thing I haven't talked about here is that when viruses duplicate their nucleic acid, their genetic information, they always make mistakes. And I think in a previous class, someone asked me how often do they make a mistake. Every time they duplicate their nucleic acid, they make an error. So these errors actually lead to different strains of viruses because they can cause changes in the viral proteins. So, for example, HIV in a person in the course of a day makes thousands and thousands of mistakes, and the viruses are different. They're all different, and some of those can eventually become a strain. And that's also the case with influenza. So every year we need a new influenza vaccine because the virus makes mistakes, and the ones that help the virus to grow better are the ones that show up next year. Okay, another one? First name?
Ariana. If, since they make mistakes, they make different strains, wouldn't there be more than just one different strain every year?
Yeah. In fact, there are different strains all over the world. That's picking the one for the vaccine is a tough choice every year for flu. And we don't always pick the right one. So flu in the winter is up in the northern hemisphere. And then when it's summer here, it goes down south, which is their winter. So what we do is we look at the strains that are circulating in the southern hemisphere. We pick one of those for our vaccine for the next winter because air travel brings viruses back and forth pretty quickly. But we don't always pick the right one because you're right, there are many, many different strains throughout the winter. So you have people working pretty frantically in virology labs to figure out which one to go. They try and see which one is the most predominant, but they don't always get it right. Absolutely. Anything else? One more question? One more. You did a great job. All right, I'm done. Thank you very much. You were Lauren Dooley, right? And you're a biology teacher at Scotch Plains Fanwood High School. So I just spent the day teaching your classes about viruses. Do you think this is a good thing at this level of biology for them to hear?
Absolutely. I think it's really vital for them to understand how the science is not just isolated to their classroom and how it is something that's very relevant to their lives and will continue to be relevant, and that it's a changing thing because they oftentimes, I think, forget science is a process and that we ask questions and we don't really know things. And they often look to me and say, oh, well, you know the answer, give me the answer. And I'm like, no, I don't necessarily always know the answer. And I think today was really good because they ask a lot of questions and a lot of your responses was, well, we're not really sure. And that's good for them to see that you're someone who's been in the field for a very long time and you've been studying this particular organism or not. And, there are still unanswered questions. And so I think that's really important for them and it is relevant. And so I think it was something very useful for them.
One of the things I struggle with is there's a big gap between what we do, research scientists, and the rest of the population. And I try and do outreach as much as I can to teach, but I can't reach everyone. Is this something that you see? Do you see a gap between what the kids understand and maybe what their parents understand? And if so, how do we take care of that? What can we do?
I think there is a gap because they come in and they don't really know what happens in science. And I think they have a skewed vision, even though we try to tell them that it's not lab coats and sitting by yourself and big Einstein hair. They still seem to think that. And so a lot of them are less excited about going into the field or exploring the fields. And I don't think they even realize how broad science is. They see it as the three sciences that we cover here, and they don't realize that biology is many things, chemistry is many things, and you put them together, and there's a lot going on there too. So I think that by having someone like you come in where they can see something practical going on in the field, that's useful. I think what you do with the podcast is awesome because that's connecting to them in their world with the technology, because we all know that at this age they're living in the technological world. And I think even things where if they could visit a lab, I'd love to get the kids to go, maybe a field trip at some point out to these labs, that kind of outreach on both ends where, you come here, we could come out to you or other labs so that they can see where it is in the world and how they might get involved. Because I think not everybody's parent or guardian is a scientist or involved in science, and they have those same misconceptions as their kids, and it just keeps cycling. And I think that we need to show them there's a lot more out there than they even know about.
So you're welcome to come to my lab with your class anytime. We could set up a day. They could visit. I could have some of my colleagues give them little presentations. They could see what a real lab is. You're welcome to do that if you want. It's in New York. It's kind of a chore to do it, but you're welcome to do that. So I was inspired by a biology teacher in high school. That's why I went into science. Can you see sparks in kids that would make some more likely to go into science than others?
I definitely see sparks in them, and what's nice at this age is there's little sparks in all of them, and so I try to really find it. That's the tough part, is finding what is going to create the spark in each one of them, and then I try to get them excited about anything that I can. So they're definitely, in each of my classes, I can see kids that are excited, and as long as we don't dampen that spark, I can see it growing. And so that's what I try to do all year long, is foster that excitement and energy, because I think that's the most important thing. Because I found in my experience that teachers that weren't as enthusiastic or just confused me made me not want to go into different areas. And those that were exciting and that kept asking me, are you interested in this? Why don't you pursue it? Then I went towards those subjects. So I try to grab onto the ones that I know there's something, and I say, all right, let's find the things that are going to make you excited.
There are also some kids who clearly are not interested. So what do you do with them? How can you capture them?
I found that being interactive in here is the best way to get everybody motivated on some level. So I've really worked very hard to change up the way I teach and make everything in some capacity. Group work, lab work, simulations, making posters, because they have so many different talents. Some of them are my writers, some of them are actors, some of them are presenters, some of them are going to be the drawers. And if you can tackle a little bit at a time, maybe not everybody's going to love every project or every assignment, but to get them interested in something, it definitely helps them, I think. And the last few years, I've kind of surveyed my kids at the end, and almost all of them, not always all, but the majority have said, you know what, I don't love science, I'm probably not going to go into it forever, but this was fun, and I could see why people do it, and I understand it better now.
The format of what I did, I think, is not great, to have a PowerPoint and just stand up and talk, because I think it's all I could do to introduce them, but I wouldn't want to do that on a daily basis. I can see that walking around and having them do things would be much better. So why did you decide to teach biology rather than, say, do research in the field?
I think from a very young age, I always kind of had that nurturing, teaching character. I was the 5-year-old teaching my 8-year-old friend how to do multiplication. So it's just part of who I am. And I went into a lab for an internship and found that I was more lonely, and it was probably that particular lab setting. If I had a different experience, it might have gone a different way. But a lot of people around me were going into education. I said, it's something I always wanted to do, and science is what I love. So I came in and I love it every day. I love it because I get to work with these kids. And I heard you say before, it's the questions, and they always have questions, and they always have different thoughts, and they bring something new to the table every day. And that is so fun to watch, because I get to change up what I do based on them, and every day we learn together. And so it's still a learning process for me, and I think it's great.
Yeah, I think the questions are the best part. That's one of the reasons I wanted to do this, because those who know the least always ask the best questions. So what's the best part about teaching here?
Gosh, that's a hard question. There's a lot of best things. Honestly, it's the kids. It's the kids, because I could have had a terrible evening at home, stressed, this, that, but it doesn't matter, because when I walk in, they come in, hi, Miss Dooley, and they have energy that just lights me up, and it just fires me up to do what I have to do, and then we just feed off each other, and it's just every day there's energy in the class, and that brings me back day after day.
So you think you'll be doing this your whole career, or do you have other ideas for the future?
I'd love to continue educating myself, but always probably in a situation where, whether I'm here, there, or somewhere beyond, it'll probably be in some sort of teaching capacity, because I just love doing it.
Thanks so much for talking to me, and thanks for having me today. I really enjoyed it.
Oh, thank you very much for coming. I think it was a wonderful experience.
Before we wrap up this episode of TWiV, I'd like to read a few reader emails, because it's always good to catch up on the long list that we have. Andrew wrote, Dear TWiV, I've been listening to your TWiV podcast since episode 15 or so, and have found it to be the best podcast out there. I'm an environmental microbiologist and pollution scientist, and found your podcast a great way to get a better understanding of the complementary discipline environmental virology. More to my question. How long does flu immunization last? I assumed it was for life. It was also my understanding that it effectively only provides 1 to 2 years protection, only because the virus is rarely around in a sufficiently similar form to afford immunity longer than a few years. Having said that, I've seen a table somewhere indicating that the vaccine only affords 1 year immunity. How does this work? Okay, he's got 2 other questions, so I'll answer that one first. Well, natural infection, of course, lasts a lifetime, because we have a good memory of our initial encounter with the virus. Now, the vaccine is shorter. It depends how old you are. If you're younger, your immunization lasts longer, but in the older individuals, it may last only a year. So maybe that's what you're thinking about when you indicate that the vaccine only affords a year immunity. Now, of course, you're right. The strains do change every year. So even if the memory of a vaccine lasted longer, it would not be terribly effective, because you would need a new vaccine to counter the new strain. So in effect, practically speaking, the vaccine's only of value for a year or two, but the memory in younger individuals probably lasts at least 5 to 10 years. His second question is, if there is limited immunity with the influenza vaccine, why do other vaccines for other viruses afford lifetime immunity? Is it both a function of the virus and the vaccine? If part of the answer is that the vaccine does not stimulate the immune system effectively, is this not correctable? Is the motivation to vaccine manufacturers just not there, or is the science not there? Well, it's a complicated question, Andrew. We don't understand, first of all, why older people don't have a longer memory to influenza. We just don't understand the mechanistic reason for that. A second reason for the shorter duration of memory is probably because it's a disrupted vaccine. It's inactivated and doesn't completely mimic a real infection. For example, the polio vaccine, one of the polio vaccines, which is infectious, affords lifetime immunity because the virus actually replicates in you. So in general, infectious vaccines will create longer enduring immunity than inactivated or disrupted ones. It's not unusual, for example, for those who received the inactivated polio vaccine in the 50s to have lost their memory responses by the 70s or 80s. With newer production methods, it's possible to make inactivated vaccines that have a higher antigen content and which produce more enduring memory. But we haven't yet done that with influenza vaccine. Now, I don't think the motivation of vaccine manufacturers is an issue because they get to sell vaccine every year anyway. There are always new people born, and so I don't think that really motivates them one way or the other. I think they're always trying to find new ways to develop new vaccines, and we've talked about some of those on TWiV, such as the plant-grown subunit virus vaccines for influenza. From a mechanistic perspective, what is the difference to the body by having flu mist versus injection? From your previous discussions on this topic, it's clear that the body sees the vaccination differently, as flu mist provides a true infection while the injection does not. If the infection method is more realistic, does it afford longer immunity than the injection? This is obviously linked to my previous question. Well, you're right, flu mist mimics a natural infection because the virus is infectious. In theory, it should provide longer-lasting memory. And the vaccine hasn't been out long enough to really test that, but it's something that I'm sure we'll be looking at. It's a bit of a difficult question to answer because if you, say, in 10 years' time, try to see if there's still a memory response to flu mist, it may be difficult to distinguish that from a natural infection. So those are difficult studies to carry out. But in theory, it should last longer. In practicality, again, because the virus changes, it's not useful for protection after a year or two. Thank you very much for your time in answering these questions. I'm sorry it's about H1N1 influenza, as I know from your latest podcast you're itching to speak about something else. Well, I don't mind talking about it. I realize that's what everyone is interested in this year and probably for the next year as well. And I'm happy to talk about it because that's part of our mission here at TWiV is to clarify issues that are of importance to people with respect to viruses. James writes, "Hello TWiV, I came across your podcast recently in a science blog and wanted to thank you for putting it together. I've just graduated from college with a degree in physics and I'm taking a year off to work in Japan and learn how to talk about science in Japanese before going back to the US for a PhD program in biophysics. I knew almost nothing about viruses before I started listening, but now I'm considering focusing on viruses when I return. I had two questions that I thought you might be able to shed some light on. The first is about a practice here in Japan known as ugai, which is translated in English as gargling. In addition to washing hands and using alcohol-based disinfectant, Japanese people are taught at a young age to gargle with either water or a solution of povidone iodine, which seems quite strange to an American. I work at a school, and there's been more encouragement to gargle because of concerns over swine flu since the fall term started. I am a bit skeptical, although I think it can't hurt, and was wondering if you thought there was any way this could prevent infection, either by removing some mucus from your upper throat or by the use of povidone iodine. I'm still catching up with your podcasts and haven't yet reached your discussions on swine flu, but I'm guessing not because influenza is often transmitted by aerosol. My second question is how you all have balanced breadth and depth in your scientific education. That is, how you balance time spent focusing on your specific field versus learning about related or seemingly unrelated science. I find myself wandering far and wide in what I read, and was wondering if you had any advice for someone entering their graduate studies. Thanks again, and sorry for the long email. All right, about the gargling. Well, removing mucus isn't a good idea, of course, because mucus helps prevent viruses from reaching the respiratory epithelium. On the other hand, any removal is bound to be minimal and transient as the mucus is regenerated. So I doubt that would make any difference one way or another. So what is gargling doing? All right, so it might remove some viruses that you've recently inhaled, but the effect will wear off shortly, and then you can inhale new viruses. It's not going to last a long time. So my view is that it's quite useless. It may make your breath smell better, but it will provide very little. It will provide very little protection, in my view, against infection with influenza virus. Now, I noticed that there are several publications in PubMed which in fact deal with gargling in Japan to prevent upper respiratory tract infections. So here's one from BMC Health Services Research from a Japanese group. In Japan, gargling is a generally accepted way of preventing upper respiratory tract infection. The effectiveness of gargling for preventing infection has been shown in a randomized controlled trial that compared incidences of upper tract infection between gargling and control groups. Now, they don't have that trial in this paper, and I haven't looked at it, but it's just one trial, and I'm not sure of the how effective it was, in fact. Another study in dermatology shows that a number of viruses that are human pathogens can be inactivated by povidone iodine, and that's great, but again, the inactivation is going to be transient. So my feeling in general is that this is not a terribly effective way to prevent upper respiratory tract infections. Bill writes, I continue to be hooked on TWiV. Now I start checking Sunday afternoon for each new episode. My twin regrets are that, one, I seldom am able to listen a second time to the casts, and that therefore much of the detail is lost to me, and that, two, at least so far, neither of my daughters seem to be moving into biotech or virology, where I keep insisting all the cool stuff is happening. One is at the U of Michigan in an engineering program, the other just beginning to evaluate her collegiate options. She likes New York and did really well on her ACTs, hint, hint. The cool stuff. Remember me, the virus, as a cancer cure guy. At the risk of being annoying, let me bring you up to speed. Oncolytics, the Canadian company, recently reached agreement with the U.S. FDA to begin a Phase 3 trial of its anti-cancer agent, a live, fully replication-competent virus named Riolysin. There seem to be lots of studies preparatory to the human trials, and I have attached one paper which demonstrates the synergistic effect of Riolysin used in combination with frontline chemotherapeutics. I've not been able to understand very much of the paper, but it seems to suggest that while Riolysin, Gearing type 3 reovirus, works somewhat against 7 of 9 non-small cell lung cancer types, in combination with the chemo it works strongly against all 9. And there are some really cool pictures that I think I do get, especially the picture of one cancer cell, a target of the dual therapy, dark, deflated, and lifeless. It is amazing to me that it is even possible to take pictures of virus particles inside cells. That process in and of itself would make a satisfactory podcast for me, but you already know what I am pining for, a full-fledged discussion of how this discovery was made, the path forward, ever hopeful toward an approved therapy, and why this particular virus is able to grab hold of a tumor cell's reproductive machinery and replicate. Patiently yours, Bill. Bill, we haven't forgotten. I know just the person or people to try and join me on this show, and they're a bit evasive, so I'm working on it. There are, as you know, a number of viruses that are being used in trials of therapy for curing tumors. Reovirus is just one of them, and we're toying with having episodes on each one individually or a review episode on all of them. But it will come forward, I promise you. And Bill sent along a reprint of this article, which I'll attach at TWiV.tv. Carl wrote, Hello, I have a question regarding the recent spread of H5n1. In Sweden, it has not been very widespread and seems to spread slower than expected. Now, one newspaper article claims that some MD says that they think rhinovirus has competed out H5n1 virus, in that a cell cannot be simultaneously infected by both viruses. Is this really true? This was written in the respected Swedish newspaper Svenska Dagbladet. Yours sincerely, Carl. And along with this is a second email from Andreas, who wrote, I would like to begin by saying thank you for an excellent podcast. I'm a Swedish software developer myself, so the content is quite challenging, but I still feel like I learn infinitely more from listening to an intelligent and fair discussion between experts than the slanted and shallow reporting you get in the popular media. Now, on to my question. In Swedish media during the last few days, I've read several reports claiming that one of the reasons the H5n1 pandemic hasn't really spread very well yet has been due to competition from the common cold. After following your podcast, it was my impression that the only reasonably well-supported limiting factor for the spread of influenza was absolute humidity. After some digging, I managed to track down the source of this claim, which seems to be a small study done by a researcher at the Swedish equivalent of the CDC. Reading the actual publication, though, they are much more cautious in their conclusions than the general media. Quote, the report is written mainly to stimulate interest and research concerning the possibility that viral interaction may affect influenza epidemiology. But their hypothesis seems to the best of my understanding, to have been fairly described in the general media. It would be interesting to hear your take on their hypothesis and what, if any, practical implications it might have if it turns out to have merit. Keep up the good work. And he sends a copy of the article, which is in Eurosurveillance. It's called Does Viral Interference Affect Spread of Influenza? And it's about rhinovirus epidemics occurring after the start of school, which may interfere with the spread of influenza during the period when warm and humid climate decreases the flu spread by aerosol. And as far as I can tell, there is some epidemiological information for this in the paper in terms of surveillance in Sweden. But there's no evidence that their hypothesis is correct. For example, they look at virus isolation from individuals with upper tract infections. They look at rhinoviruses isolated and influenza viruses isolated. But the real question is, would rhinovirus interfere with influenza virus? And we don't know the answer to that. As far as I know, this isn't something that's been studied. And it's really an interesting question. And it's actually got me to thinking of whether I can do an experiment to address this. And so, in fact, what should be done initially is to simply take cells and infect them with both a rhinovirus and an influenza virus, and to see if influenza will replicate in a cell that's already infected with rhinovirus. Now, rhinoviruses alter the cells in ways that could interfere, theoretically, with influenza virus replication. So if you first infect with rhinovirus, which would be the scenario in people, is influenza unable to infect those cells? So that's something that I think I might pursue in my laboratory. But as far as this study goes, it seems to me the only evidence is some observational information on a small scale in Sweden. There could be other factors at play as well. So while it's theoretically possible, it's not supported yet by any data. But it's a good idea to get the hypothesis out there and stimulate some work, and that's what it's done for me, in fact. I ought to say that the idea that the H5n1 strain hasn't spread all that much is probably a combination of seasonal influences like humidity and temperature, but also immunity of the population. And in particular, those born earlier in the 20th century, seem to be more resistant to the virus, probably because they have had more influenza experience. And I've written about that at Virology blog in a post entitled Older Age is a Good Protection Against H5n1 Influenza. So you might want to have a look at that. So that will do it for another episode of TWiV. I want to thank Miss Dooley for arranging my day at the high school, and also Miss Canfield and Miss Davis for their enthusiasm. Many thanks also to the students who sent wonderful thank you cards, which I've posted at TWiV.tv. I'd be happy to teach virology to local high schools. If you're a biology teacher and you're interested, contact me at TWiV@TWiV.tv. As always, we love to get your questions and comments. So you can send us an email, twiv@twiv.tv. You can send us an MP3 file, or you can call us on Skype, TWiV Podcast, and leave a voicemail. If you're a new listener to TWiV, please consider subscribing in iTunes. It's free and lets you receive each new episode as it's posted. If you don't use iTunes, you can always download episodes at twiv.tv, where we also have show notes. If you like TWiV, please tell others about it. Links and reviews in iTunes are particularly helpful. TWiV is part of sciencepodcasters.org and promednetwork.com, websites where you can find high-quality science podcasts from a wide variety of fields. Thanks for joining us. We'll be back next week. Another TWiV is viral.